Hello everyone. This video will be about cells, cell organelles, and I gave it the name of cell machinery because it's not just about the list of organelles and what do they do. It's about how do the organelles work together to have that cell be able to sustain its life, and most importantly, to produce and use the proteins. It all goes back to the proteins. Before we start, just a review, which is the basic stuff: prokaryotes versus eukaryotes. This is very basic. The prokaryotes are the cells that are still primitive. They look like all they have inside them is a DNA, circular DNA without a nucleus, and a cytoplasm with few ribosomes, and they have a cell wall outside the cell membrane. As for the eukaryotes, these are the ones that are complex. They have a lot of organelles, they have a nucleus, they are bigger, they are more advanced, they came later. So you can find the differences between prokaryotes, eukaryotes anywhere. But what we'll be talking about now is the eukaryotes, since they are the ones that have an actual complex machinery inside of them. I'll be using this picture which shows a eukaryotic animal cell to explain about all the parts. Now, if it's all, as I said, it's all about proteins and how do we make them, how do we use them. What builds proteins are these dots here, which are even found in the cytoplasm, which are the ribosomes. So if I want to show these dots that are on the rough ER are called the ribosomes. And as you have seen before, if you should get the list of organelles and what do they do, I will not go over that. I'm just talking about how do they all come in together. So the ribosome builds proteins, but if we want to start with the story of making proteins, that doesn't start here. The ribosomes, we should get the recipe of how to build proteins, which is how to link amino acids after each other, from somewhere, which is of course the DNA inside the nucleus. This part here in the nucleus, which is shaded, that's the nucleolus. The nucleus is just very dense chromatin, which is DNA and protein, and this is where the ribosomes are made. As for the rest will be the chromosomes, depending on which organism this is, how many chromosomes we have, and the DNA. The substance is called chromatin, and which is DNA wrapped around proteins called histones. Now the nucleus, as you see here, has pores to allow things to move out and in. The DNA will not go out for the ribosomes to read it. It will send copies, and these copies are the RNA, and to be more specific, they are the mRNA, the messenger RNA. The messenger RNA are the copies of the genes of DNA which will be sent to the ribosomes. The ribosomes will read these copies and build proteins. Now the ribosomes are located on these structures. This structure, which is the endoplasmic reticulum, and here it's called the rough ER. Rough endoplasmic reticulum, because it has the ribosomes on it. The other parts which do not have ribosomes, they will be the smooth ER, and they take care of lipids, managing lipids, production of lipids, storing calcium. But what we care about now is the ribosomes. Now if I want to magnify this, so let's say this is the surface of the rough ER and this is one ribosome. The ribosome will build a protein, which is a chain of amino acids linked together directly into the rough ER. So the protein will be inside the rough ER here. Now the rough ER, what does it do? It puts that protein into a vesicle. So I will have like a bulge here, which is similar to exocytosis, and then forming a vesicle having the protein in it. So from the rough ER, we will always have vesicles coming out to all different parts of the cell, and they have proteins inside them. These proteins were made by the ribosomes. Now these proteins are still immature. they can't do the job they have to do. Some changes should happen to them, which is in the 3D shape. So we mentioned before that the proteins have a specific 3D shape which will allow them to do their job, like enzymes, active site, etc. To acquire that 3D shape, they should be packaged into a specific way, and the organelle that packages and sorts protein is the Golgi apparatus or Golgi body. So the vesicles will go to the Golgi, and the Golgi is made up of several sacs, so it will go in from here, starts going from one sac to the other, and then exits from here. And now throughout its passage through the Golgi, the protein is packaged, it's in a 3D shape, it's mature, now it's ready to work. What will happen to the protein next? It might go outside by exocytosis. If its role was outside, let's say this is a pancreatic cell releasing insulin, this is insulin, it will be released outside, or it will be sent to the inside if maybe the role is an enzyme inside the cell. Or it has any structural role inside the cell. So it depends. It might go outside, it might stay inside. Now, we have ribosomes in the cytoplasm, and these ribosomes would create also proteins, but these proteins will not be put into vesicles, so these proteins would most probably be used directly in the cytoplasm and do not need any modification. The whole process we've mentioned here requires energy. That energy comes from the mitochondria, which is here, which provides ATP to fuel the whole process. And the movement of the vesicles usually happens on cytoskeleton, which originates from the centriole. So this is the centriole making the microtubule part of the cytoskeleton, which also is the spindle fiber, which is we see in mitosis and meiosis. So cytoskeleton, other than keeping the shape of the cell, they help in the movement of vesicles, even of whole organelles. And to get these raw materials, which is the amino acids for the ribosomes to build, we get them from the nutrients usually, which come in by endocytosis, also by vesicles. Sometimes these nutrients are complex, need to be digested, so they would fuse. Let's say this is a lysosome, they would fuse with a lysosome, which will contain digestive enzymes, which will digest these proteins or polypeptides to have the simple amino acids brought to the ribosomes for use to build a new protein. What else can we have? Vacuoles, for example, here, or any vacuole are storage of nutrients and water. So if we have excess nutrients, excess water, we store it in vacuoles. We would have several vacuoles throughout the cell. Now this is an animal cell. If I want to make it a plant cell, I would change three things. First, I would have one vacuole which is big, and everything else will be squeezed to the side, so the nucleus even will be squeezed to the side, and the vacuole will fill most of the cell. I would add chloroplasts. Usually we show them like this. These are the thylakoids, like coins, and that look green usually. And I would add outside a cell wall, and we'll have the shape of the cell determined square or rectangle. This is what I call the cell machinery. Of course, the stuff go in and out by the cell membrane. Hopefully I didn't forget any important organelle. We have some organelles, for example, called peroxisomes, which acts like a detoxifier. They detoxify toxic stuff. And before I finish, few words about the cell membrane. The cell membrane, you all know, is made up of phospholipids, and we show phospholipids as head and tail. The head is the phosphate, and the tails are the fatty acid tails. The phosphate is hydrophilic, and the fatty acid tails are hydrophobic, so they're afraid of water. That's why they line up in this specific shape, where the tails are to the inside and the heads out to the outside, because we have water outside the cell membrane and inside. So if this is the inside, this is the outside of the cell, we would have water everywhere. But here in this region, we will not have water. That's the hydrophobic area. We do have proteins such as integral proteins embedded through the cell membrane. So here we have the continuation of the cell membrane. We do have extrinsic proteins, intrinsic proteins. We do have carbohydrate chains attached to proteins, which will make them cause these to be glycoproteins, or attached to the lipids, which will cause this to be a glycolipid. We do have cholesterol, which causes the cell membrane to be more rigid in the middle of the hydrophobic area. Some proteins act as channel proteins, so they would be like this to allow stuff in or out. Some proteins will act as enzymes or receptors, or for ligands and hormones, etc. And this is about the cell membrane. Of course, there are more details about the function of the proteins, but this is the overview, and this is the overview of the cell machinery. I hope this video is helpful, and thank you for watching.